All right, so this is part 2 of the prokaryotic structures. I've got a list here with the rest of the some that can be associated with some bacteria. Not all bacteria will have these structures. All right, so the flagella we've already talked about. Pili and fimbriae are actually going to be somewhat related to flagella. They're going to be structures that are going to come off the surface of the bacteria. Fimbriae, I know this is going to kind of look like what I drew with that peritrichous flagella. These are not flagella. The fimbriae are going to be short projections that come off of the bacteria, and what the fimbriae are involved in is going to be adhesion, so sticking to things. The bacteria Neisseria gonorrhoeae, which is going to be the bacteria that causes gonorrhea, has fimbriae associated with it. Those fimbriae can attach to the epithelial cells associated with the urogenital tract on a person and allows that bacteria to kind of sit there and now gather a place to stay and take hold and start dividing. All right? So fimbriae kind of allow bacteria to adhere to surfaces. They are much, much shorter structures than the flagella would be. All right? The other structure that can be associated with some bacteria and not all, primarily this structure is going to be associated with gram-negative bacteria. And that's going to be the pili. And we'll talk a little bit more in detail about what the pili does specifically a little bit later. But what the pili is it's essentially, you can think about it as a bridge that helps connect. I'm going to draw another bacteria over here. It's going to be shorter. Don't mind that. Okay? Essentially, it's going to connect two bacteria together. And what it's going to do is it allows DNA to travel from one bacteria to another. So it's kind of a hollow tube that allows for the transfer of DNA. Typically, the DNA that's going to be transferred there is what's known as a plasmid. That plasmid's going to be a short, round, circular bit of DNA that isn't essential to the bacteria, but confers some advantage. A lot of antibiotic-resistant genes travel on plasmids, and that's how antibiotic resistance is transferred from one bacteria to another. Okay? And the structure that's going to happen by is going to be via the pili. Okay? Our next structures that are going to be kind of related, all right, so we can kind of group flagella, pili, fimbriae. Those are all a little bit similar. The capsules and the slime layers also a little bit similar, all right? We're essentially talking about some kind of, usually sugar coating on the outside of the bacteria, all right? And in terms of that, if a bacteria has a capsule, it is usually going to be something that's going to be a pathogen, meaning it can make you sick, all right? That sugar coating associated with the capsule, all right? And I'll draw that as kind of another pink squiggly line around the outside here. We're getting kind of full. What that capsule does is those sugars essentially aren't going to be something that are going to trigger your immune system. So having that sugary capsule on the outside of the bacteria essentially allows it to escape phagocytosis by the immune system. So the bacteria essentially are cloaked or hidden within the body, and that allows them to survive and multiply and therefore make you sick. Okay? So capsules are associated typically with pathogens. All right? Slime layers are a little bit different. They're a little bit more watery than a capsule layer would be. And a slime layer is going to be involved in more protecting the bacteria from dehydration. So the slime layer is there to kind of protect the bacteria from the elements, from all of the cytoplasm leaking out. All right? So those kind of go together. Our last groups are all a little bit different from one another. Inclusions. I'm going to draw this here as an inclusion in pink. Are regions within the bacteria where the bacteria is storing something. All right? It's not an organelle. It's not surrounded by a cell membrane. All right? But those inclusion bodies typically are going to be where certain bacteria will store certain substances. There are some bacteria that actually store iron in them. And so iron can be one structure that you might find within those inclusion bodies. There are many, many other things that can be stored. It depends on the type of bacteria, what's inside that inclusion body. But what the inclusion body looks like when you stain the bacteria, it looks like a dark area within the cell, okay? And in terms of that inclusion body, it's going to be associated with storage, all right? I'm going to kind of skip actin cytoskeleton here for a minute because I want to make sure to talk about endospores, okay? And in terms of those endospores, lots of times students, when they're reading through the textbook, they're like, oh, endospores, that has to do with reproduction. No. Endospores are not a reproductive structure associated with bacteria, all right? Endospores allow for survival, okay? What those endospores do is they form a protective coat around the bacterial DNA so the bacteria can withstand harsh conditions. And when we're talking about harsh conditions, it can be a number of different conditions. Lack of water would be a great example of a harsh condition. High temperatures would be an example. Caustic chemicals would be an example that would cause those bacteria to form an endospore. And I'm going to kind of draw this one a little bit separate from what's over here, okay? Because you'll have the bacterial DNA here. And essentially what the bacteria will do is it's going to kind of partition off a copy of that DNA, all right? So we'll label this with DNA here. We'll label this with DNA here, all right? What it's going to do is now it's going to put a protective coat around that DNA, all right? So there are going to be layers and layers and layers of protein and other substances that are going to essentially protect that DNA from whatever the harsh environment is, all right? And in terms of those endospores, what that does is that allows the bacteria to survive long term, all right? We're going to talk a little bit more in detail about endospores because these are really kind of an important structure. Some bacteria that form endospores are of the genus Clostridium. So Clostridium botulinum, if you've heard of that one, that causes foodborne illness, causes botulism. Clostridium tetani causes tetanus. And another one that you probably have heard of if you've already been in a healthcare setting is Clostridium difficile, or C. diff, maybe the name you know it by. All of those have the ability to form endospores. And the reason those are hard for hospitals to get rid of are because of that protective coating, okay? That allows it to withstand the harsh chemicals that are used to try and clean it up, and allows that essentially DNA to be dormant until the conditions are right for survival of the bacteria again. And the conditions that are right are when a patient touches that spore and then rubs their nose or sticks their finger in their mouth because they're eating, and they transfer the spore into their body. Your body is a nice humid environment, allows those spores to grow, hatches, allows that bacteria to multiply, and allows essentially the person gets sick. Okay? So in terms of that, the summer here, the actin cytoskeleton, I'm just going to real quick, there's an image on the PowerPoints that shows actin. Essentially what you would see, I'm going to kind of draw it around the outside here on this one. Some bacterial cells, not all, are going to have actin associated with it. And in terms of that, that's one that's really pretty rare as far as finding. All right? But it can be a way to identify certain types of bacteria. All right? Thank you for your attention. If you have any questions, please let me know.